Today we are going to learn some aspects related to complement system, classical, lectin, alternative, MAC, and hypersensitivity. Some aspects related to immunology. So complement system. The complement system refers to a series of proteins circulating in the blood and bathing the fluids surrounding tissues. So the complement system refers to a series of protein circulating in the blood and bathing the fluids surrounding tissues. The proteins circulate in an inactive form, but when activated, they act in concert in an orderly sequence to exert their biological effects. The term complement refers to the ability of these proteins to augment the effects of other components of immune system. For example, antibody. So pathways of complement activation: classical, lectin innate, alternative innate. Okay. Classical in the sense AB binds to specific AG on pathogen surface. Then lectin innate, mannan binding protein binds to mannose on pathogen surface. Then alternative innate, pathogen surface allows complement activation. Then complement activation, recruitment of inflammatory cells, opsonization of pathogens, lysis and death of pathogens. Classic pathway: antigen antibody complexes, minor anaphylaxotoxin, kinin-like activity, anaphylotoxin, chemotactic factor, C3 convertase, C5 convertase, stabilized by polysaccharides and bacterial cell wall, alternate pathway. Then terminal components, classical pathway, generation of C3 convertase. The binding of C1 to antibody is via C1q, and C1q must cross-link at least two antibody molecules before it is firmly mixed. The binding of C1q results in the activation of C1R, which in turn activates C1s. The result is the formation of an activated C1QRS, which is an enzyme that cleaves C4 into two fragments, C4A and C4B. The C4B fragment binds to the membrane, and the C4A fragment is released into the microenvironment. Activated C1QRS also cleaves C2 into C2A and C2B. C2A binds to the membrane in association with C2B, and C2B is released into the microenvironment. The resulting C4B C2A complex is a C3 convertase which cleaves C3 into C3A and C3B. C3B binds to the membrane in association with C4B and C2A. C3A is released into the microenvironment. The recruiting C4B C2A C3B is a C5 convertase. The generation of C5 convertase is the end of the classical pathway. Biological activities of classical pathway components: component C2B, prokinin cleaved by plasmin to yield kinin, which results in edema. That is the biological effect. Then C3A, anaphylotoxin, can activate by basophils and mast cells to deregulate, resulting in an increased vascular permeability and contraction of smooth muscle cells, which may lead to anaphylaxis. Then C3B, opsonin, activation of phagocytic cells, anaphylotoxin. C4B, opsonin. Then lectin pathway, mannose binding related, initiated by the binding of mannose binding lectin MBL to lectin pathway, mannose binding related. So initiated by the binding of mannose binding lectin MBL to bacterial surface with mannose containing polysaccharides. Binding of MBL to pathogen results in association with two serine protease, MASP1 and MASP2, MBL associated serine protease. So initiated by the binding of mannose binding lectin MBL to bacterial surfaces with mannose containing polysaccharides. Binding of MBL to a pathogen results in the association of two serine proteases, MASP1 and MASP2, MBL associated serine proteases. Formation of the MBL, MASP1, MASP2 trimolecular complex results in the activation of the MASP and consequent cleavage of C4 to C4A and C4B. The C4B fragment binds to the membrane, and the C4A fragment is released into the microenvironment. An activated MASP also cleaves C3 C2 into C2A and C2B. C2A binds to the membrane in association with C4B and C2B is released into the microenvironment. The recruiting C4B C2A complex is a C3 convertase which cleaves C3 into C3A and C3B. C3B binds to the membrane in association with the C4B and C2A and C3A is released into the microenvironment. The resulting C4B, C2A, C3B is a C5 convertase. The generation of C5 convertase is the end of the lectin pathway. Bacterial protein and polysaccharide with terminal mannose residues, formation of C3 convertase, MBL mannose binding lectin, MASP mannose associated serine protease. This is the lectin pathway. The lectin pathway is initiated by binding of lectin such as mannose binding lectin to microbial surface carbohydrate. MASP2 is structurally related to serine protease C1s and can cleave both C2 and C4, giving rise to the C3 convertase. Then alternative pathway. Activation of the alternative complement pathway begins when C3b or C3i binds to the cell wall and other surface components of microbes. C3b can also bind to IgG antibodies. Factor B then combines with cell-bound C3b to form C3b beta B. So factor D then splits the bound factor B into BB and BA, forming C3b BB. So properdin then binds to the BB to form C3b capital B small b B T that functions as a C3 convertase, capable of enzymatically splitting hundreds of molecules of C3 into C3A and C3B. So C3 is an unstable molecule continuously undergoing hydrolysis in the blood and lymph. This rate of hydrolysis can accelerate in the presence of some pathogens. So C3b then binds to factor B, making it susceptible to cleavage by factor D. This creates C3 convertase of the alternate pathway. C3b capital B B. Then activation of C3 and C5. Central molecule in the complement system, highly labile, then present in large amounts in serum, after which activation of the alternative complement pathway begins when C3b or C3i binds to the cell wall and other surface components of microbes. Alternative pathway protein factor B then combines with cell-bound C3b to form C3bb. Then some of the C3b subsequently binds to some of the C3b capital B small b to form C3 small b capital B small b 3b. A C5 convertase capable of enzymatically splitting hundreds of molecules of C5 into C5a and C5b. Then the MAC, the membrane attack complex causing cell lysis. The C5b6789 membrane attack complex puts pores into lipid bilayer membranes of human cells to which antibodies have found. This results in cell lysis. MAC can also damage the envelope of enveloped virus and put pores in the outer membrane and cytoplasmic membrane of gram-negative bacteria, causing their lysis. Biologically active functions: cell lysis, inflammatory response, chemotaxis. Once again, complement component: cell lysis, C5b9 membrane attack complex, inflammatory response, C3a, C4a and C5a, anaphylaxotoxins. Then chemotaxis of leukocytes, C3a, C5a, opsonization of particulate antigens, viral neutralization, solubilization and immune clearance. Then hypersensitivity. Hypersensitivity, also called hypersensitivity reaction or intolerance, refers to undesirable reactions produced by the normal immune system, including allergies and autoimmunity. These are usually referred to as the overreaction of the immune system, and these reactions may be damaging and uncomfortable. So hypersensitivity is an undesirable reaction produced by normal immune system, as it includes allergy and autoimmunity. So hypersensitivity, also called hypersensitivity reaction or intolerance, refers to undesirable reactions produced by the normal immune system, including allergies and autoimmunity. They are usually referred to as an overreaction of the immune system, and these reactions may be damaging and uncomfortable. So hypertension sensitivity is an undesirable reaction produced by normal immune system. It includes allergy and autoimmunity. So hypersensitivity reaction require pre-sensitized immune state of the host. There are two categories of adaptive hypersensitivities. Immediate hypersensitivity refers to humoral immunity and antigen antibody reactions. Then delayed hypersensitivities refer to cell-mediated immunity, cytotoxic T lymphocytes, macrophages, and cytokines. So Gell and Coombs classification of hypersensitivity reactions, type 1, 2, 3, and 4. Type 1 IgE mediated allergen. IgE binds to mast cells with inflammatory mediators released. Anaphylaxis, angioedema, immediate minutes to hours. Type 2 cytotoxic IgG and IgM bind to allergens on target cell, complement mediated neutropenia, some associated diseases. And type 3 immune complex. Type 4 delayed cell mediated. Type 1 hypertension sensitivity, IgE Fc region mediated anaphylactic. The reaction occurs on exposure to allergen second time. The first exposure, sensitizing dose, results in sensitization of host to allergen, and subsequent exposure, shocking dose, will cause reaction. Anaphylactic shock occurs in sensitized animals within seconds to minutes. 15 to 13 after exposure to allergen. After exposure to the allergen, now called as an allergen. Sometimes the reaction may have a delayed onset. In type 1 hypersensitivity reactions, allergens are proteins with a molecular weight ranging from 10 to 40 kilodalton. Diagnostic tests for immediate hypersensitivity include skin prick and intradermal tests resulting in beat and flare reaction, measurement of total IgE and specific IgE antibodies against the suspected allergens by ELISA radio allergosorbent test. Then mechanism. In the first exposure to allergen, IgE is produced, presumably under the stimulation of T helper type 2. These IgE bound to the Fc receptor on mast cell. The mast cell is sensitized against the allergen. Subsequent exposure to allergen results in degranulation of the mast cell, release its mediators, namely cytokines, vasoactive amines, and eicosanoids, prostaglandin, and leukotrienes. These mediators cause vasodilation, increased vasopermeability, tissue damage, and smooth muscle contraction. This is called early phase reaction. Then other mediators, especially cytokines, recruit other inflammatory cells to the site of reaction. This is called a late phase reaction. Clinical example: asthma, atopic allergy, and anaphylaxis, very rapid body allergic reaction. Then type 2 in the sense, type 2 hypersensitivity, antibody mediated cytotoxic. Type 2 hypersensitivity are antibody mediated cytotoxic reactions occurring when an antibody binds to antigens located on surface of cells, usually RBCs. Reaction time is minutes to hours. It is mediated primarily by antibodies of IgM or IgG class. The bound antibody can cause cell lysis by activating the classical complement pathway, promoting phagocytosis, opsonization, or through ADCC. Many different antigens may trigger the cell destruction, but an infection in a genetically predisposed animal appears to be a major targeting pathway. So antigens are normally endogenous, although exogenous chemicals, haptens such as drugs that can attach to cell membranes, can also induce type 2 reactions. So autoimmune hemolytic anemia, blood transfusion reactions, erythroblastosis fetalis, drug-induced hemolytic anemia, granulocytopenia, and thrombocytopenia are examples. IgM or IgG antibodies are made against self-antigens either as a result of failure of immune tolerance or cross-reactive antibodies can develop during infections, which may bind to normal tissue antigens and trigger antibody-mediated cytotoxicity. Consequent binding of these antibodies to the surface of host cell lead to opsonization of the host cell, whereby phagocytosis stick to host cells by way of IgG, C3b, or C4b and discharge their lysosomes. Activation of the classical complement pathway causing MAC-induced lysis. ADCC-mediated destruction of the host cells whereby NK cells attach to the Fc portion of the antibodies. Clinical examples: autoimmune hemolytic anemia, Goodpasture syndrome, Graves' disease, myasthenia gravis. Then autoimmune hemolytic anemia, autoimmune thrombocytopenic purpura. For example, pemphigus vulgaris, Goodpasture syndrome. Then type 3 hypersensitivity, immune complex mediated. In type 3 hypersensitivity, soluble immune complexes are formed in blood and are deposited in various tissues, typically the skin, kidney and joints. Active classical complement pathway and cause inflammatory damage. It is mediated by soluble immune complexes. They are mostly of IgG class, though IgM may also be involved. The reaction takes hours to days to develop. The antigen may be exogenous, chronic bacterial, viral or parasitic infections, or endogenous, non-organ specific autoimmunity. The antigen is soluble and not attached to the organ involved. The prerequisite for the development of immune complex disease is the persistent presence of soluble antigen and antibody. Mechanism in some people in relation to post-streptococcal endocarditis. The antibodies that are formed against streptococcus forms immune complex that is pre-deposited in vascular membrane, especially the glomerulus. This may also activate the complement system or become a target of phagocytes, causing damage to the vessel wall and ultimately the organ itself. For example, the kidney. Clinical example: systemic lupus erythematosus, post-streptococcal vasculitis, post-streptococcal glomerulonephritis. Then group in the sense type 4 hypersensitivity, cell-mediated delayed type. Characteristics involve type 4 hypersensitivity is often called delayed type of hypersensitivity as the reaction takes more than 12 hours to develop. Typically, the maximal reaction time occurs between 48 to 72 hours. It's mediated by cells that cause an inflammatory reaction to either exogenous or auto antigens. The major cells involved are T lymphocytes and monocytes or macrophages. This reaction to exogenous antigens involve T cells and also antigen-presenting cells. All produce cytokines that stimulate a local inflammatory response in a sensitized individual. So DHR cannot be transferred from an animal to another by means of antibodies or serum. However, it can be transferred by T cells, particularly CD4 Th1 cells. Mechanism: delayed type hypersensitivity mediated by CD4 plus and CD8 plus T cells. T cell mediated cytotoxicity mediated by CD8 plus T cells. In this reaction, T cytotoxic cell mediate specific for autoantigen or host cell may directly kill these cells. So APCs or tissue antigen, autoantigen active sensitize CD4 plus and CD8 plus T cells, causing them to secrete cytokines. So these cytokines recruits and activate phagocytes as well as inducing or in local inflammation. Ultimately, tissue injury occurs. For example, contact dermatitis, rheumatoid arthritis, and Mantoux test. So let's have an overview. Hypersensitivity is of two types: immediate and delayed. Immediate in the sense symptoms are manifested within minutes or hours after exposure. In case of delayed, symptoms are manifested within days after exposure. And the immediate one is of two types: ABAG mediated, AB mediated, immune complex mediated. And the AG-AB mediated is of two types: IgE mediated and IgEG or IgM mediated. Those are the two types. Then AB mediated is of one type, IgG mediated. Then T cell mediated one is involved in case of delayed hypersensitivity. So IgE mediated type 1 hypersensitivity, AG induces cross-linking of IgE bound to mast cells and basophils with release of vasoactive mediators. Then type 2 hypersensitivity, AB directed against cell surface antigens mediates cell destruction via complement activation or antibody dependent cell mediated cytotoxicity. Then type 3 hypersensitivity, AB complexes deposited in various tissues induces complement activation. Then type 4 hypersensitivity or delayed type hypersensitivity, sensitized H cells release cytokines that activate macrophage or T cells which mediate direct cellular damage. So that's all about hypersensitivity. Thank you for listening.